Hey, Bio30s, and today we're going to have our second discussion about molecular genetics, which is going to be about DNA replication. DNA is actually a very unique molecule in the fact that it's the only molecule that can actually make copies of itself without the help of any other molecule or substance. Any other molecule we need to make, proteins, carbohydrates, lipids, we all require other molecules to help make those macromolecules, but DNA is the only one that can basically, without any help, can go at it alone. It can basically make copies of itself, just basically sitting there doing its thing. So here's what I'd like to start off our discussion with today. First of all, I want to give you guys a recap of the structure of DNA from last time. Remember that we discussed nucleotides, which are the monomer units of DNA. A nucleotide is broken into three parts. In the center of the molecule, or on the innermost part of the nucleotide as it relates to the DNA molecule, we have our nitrogenous base. Now, the nitrogenous bases in this diagram would be like the little tick marks there. And just a reminder that nitrogenous bases, there's only four of them for DNA. There's A, T, G, and C. So remember that if I have an A, A's are always going to bond to T's, T's will always bond to A's, G's will bond to C's, and C's will always bond to G's. So again, T's are thymine, A's are adenine, C's are cytosine, and G's are guanine. So that's the inside of the DNA molecule. Then you go to the outside, which would be represented by that long line, which would be like your rungs of a ladder rather than the steps of a ladder. Those are going to be your sugars, your 5-carbon sugar in DNA, which is deoxyribose. And on the very outside edge, there's a phosphate molecule as well. Now, the direction in which that phosphate molecule points is going to indicate an end of the DNA strand. And the side that the phosphate molecule points to is known as the 5' end. Now, you might remember my conversation last time, I also mentioned that DNA is a double-stranded molecule, forms that double helix as discovered by Watson and Crick and Franklin. But what you might have forgotten is that it is an antiparallel molecule, so that each strand is actually in an opposite configuration beside one another. So what do I mean by that? Well, if I've got one strand that has a 5' on this end and a 3' down on that end, the strand that's beside it in that particular DNA strand or molecule has to be 3 on the top and 5 on the bottom, indicating that they are in opposite configurations of each other, otherwise known as antiparallel. Now, there is something that I want to start my discussion with today as far as the duplication aspect or the replication aspect of DNA. DNA replication is often referred to as being semi-conservative. Now, what does that mean? Semi-conservative replication simply means that for any piece of DNA that's in your body, you're looking at essentially what is one parent strand, which is represented here in blue, and a daughter strand, the one that was replicated off of the original DNA strand. So, for example, if this is my DNA that's replicating, it's basically opening itself up, it's unraveling itself. Basically, the steps of the ladder are breaking apart, allowing me to access both the strands, and then each parent strand is going to synthesize an anti-parallel complementary strand beside it. So, for example, if this one opened up on this side, here are my nucleotides and here are my nitrogenous bases being laid down. Over here, same idea. But remember, as I produce these strands beside my parent strands, my blue strands, those daughter strands have to be in an anti-parallel configuration to the parent strand. So again, this daughter strand here has to be a 5' on that end, and then this end down here would be 3' because it's closer to the 5' end of the parent strand. And over here, we would have a daughter strand with a 3' end over there. And this end, I'm going to maybe just move that over just a little bit because it might be a little confusing if I have them too close together. There we go, our 5' end. So this strand here is basically a 3 to 5, and this is a 5 to 3 strand. Now, what's the importance of this? Well, you're going to find out very quickly there's a huge importance as to the orientation of the daughter strands as they get synthesized beside the parent strands. DNA, and for that fact, RNA, when we discuss RNA later in the unit, any kind of nucleic acid, when you lay it down, when you basically build a strand of nucleic acid, that strand can only be laid down in a 5 to 3 direction. So in this case, this strand is going to be built in this direction, and in this case, that strand is going to get built in that direction. Now, they're in opposite directions from each other, and remember that because we're going to get back to that here in a few minutes, but there are some real implications as to how you can build a strand in this direction as opposed to a strand being built in that direction, which I'll get to here right away. Before we get started, I want you to quickly take a look at this little animation. This is going to walk you through the whole process of DNA synthesis or DNA replication in a few minutes. Going to give you the whole kind of three-dimensional view of what it looks like. A good video to start with before I kind of start giving you the two-dimensional diagram up on the board. It's probably going to help you out quite a bit by taking a watch of that video before I get started. All right, so DNA replication is broken into 3 stages. There is initiation, elongation, and termination. So we're going to start off with initiation and elongation first. Initiation is very simple. Initiation is simply taking that DNA strand that's, side by side, those 2 antiparallel DNA strands that make up a DNA molecule. Remember, they'd be wrapped up into a spiral double helix. What we're going to do during initiation is simply this. We're going to open them up. We're going to open up those strands and basically open up those pieces of DNA. Now, how do we do that? We need an enzyme. All of these processes that I'm going to be discussing today with initiation and elongation, each stage of these processes has an enzyme that's involved to actually carry them out. So the first stage, which is initiation, is going to basically be happening at what's called the replication fork. That's the point at which the DNA is actually being unzipped. And when you use the term unzipping DNA, it's just like it sounds. It's like a zipper on a jacket or a coat. As you unzip the zipper, you basically get your 2 openings, and then you have the section of the zipper that's still closed. That's the exact analogy you can use when you're thinking of DNA. Now, there is an enzyme that's responsible, and it's indicated by this red triangle over here. That enzyme, which is over on my key here, is called DNA helicase. Easy way to remember DNA helicase. DNA helicase, well, I'll give you the joke here in a couple seconds. It'll be probably more funny after we've had the discussion, but there's a slide, if you've already seen it, you'll get the joke here right away. Okay, so anyways, DNA helicase is going to basically open up the DNA, and the point at which the DNA is opening is referred to as the replication fork. So that is essentially initiation. Now, your notes are going to refer to things called replication bubbles. Don't worry about that. For now, just imagine that if it's a piece of DNA, as far as our conversations are going to go, imagine you've got a long piece of DNA and it's just going to unfold from one direction to the other direction, just like a zipper would. DNA does tend to have multiple replication forks because of the length of a piece of chromatin, but for our purposes, let's not worry about that at all. We're just going to assume that the DNA is unraveling in one direction, straight through from one end of the DNA strand all the way to the bottom. And by the way, all of this discussion we're having today is referring to the stage S phase. This is what's happening during S phase in the cell cycle. We talked about the different phases in the cell cycle. This is the stage where DNA is replicating prior to either mitosis or meiosis. So everything we're discussing here, this is the mechanism of what's occurring during S phase when, like, for example, in a human, 46 pieces of chromatin are going to duplicate into 92 pieces of chromatin, which will eventually become your 92 chromatids when they condense during either mitosis or meiosis. So just to give you an idea of where this is occurring, that we've already discussed. Okay, by the way, here's your joke, okay? If I were an enzyme, I'd be DNA helicase so I could unzip your genes. That's sort of the gag, because that's what, it's a bad pickup line. But that's sort of the way you remember DNA helicase. It's like a bad pickup line if I want to unzip your genes, right? And that's what it does. DNA helicase essentially unzips DNA, and that's what it does. So if that's an easy way for you to remember DNA helicase, then have at it. Okay, now we get to the most complicated part of DNA synthesis or replication. This is the elongation portion. So what we're going to do now that we've unzipped the DNA, the original parent strands of DNA, the blue strands that are the parent strands, we now have to build our daughter strands beside them. And as I mentioned, there are two ways and there are two directions in which the daughter strands are going to be built. In the case of this parent strand here, which is a 3 to 5 configuration, the daughter strand is going to be anti-parallel beside it and is going to be 5 to 3. So we know that this strand is going to build in the direction of the zipper opening. So as the zipper zips down the jacket, that daughter strand is literally building along as the zipper opens more and more and more and more. This strand is much easier to build, way less time intensive, and we call that strand the leading strand. And essentially, it's built in one long continuous piece. So it's relatively easy for the DNA to build that strand. The scumbag strand, or the dirtbag strand, the one that is much more problematic, is the other strand. The other strand that's on the parent strand that's in a 5 to 3 configuration is going to be 3 to 5, which means that it's going to build away from the replication fork. So as the zipper goes down further, it's going this way, but the zipper's going that way. So we have to build a little section. So initially here, this would have been the replication fork, and then we build a little section, but then the replication fork moves back. So I got to build another little section over here. So as this replication fork continually moves further and further away, I have to build this section, this strand has to be built in stages, in segments, in fragments. That's this scumbag strand, as scientists sometimes like to call it, or the dirtbag strand, is called the lagging strand. So the leading strand, much more easy to build. The lagging strand, a little more tougher to build, a little more enzyme intensive, as I'll get to. So what's the overall procedure? Regardless if I'm building a leading or a lagging strand, the enzyme configurations are pretty much the same. It's just how much of the enzymes are getting used. So I'm going to use my leading strand to walk you through the process, and then I'll kind of put a couple of little side notes in as it relates to the lagging strand at the end of this conversation. So what's the first thing we have to do? Well, the first thing we have to do is we have to lay down what's called an RNA primer. Now remember, we talked about RNA. RNA is single-stranded nucleic acid. It does not have a ATGC nucleotide configuration. It has AUGC, so adenine, uracil, guanine, cytosine. So we're going to lay down a very small section of RNA primer, and it's almost like a little spot where the DNA can start building. It's a part that we're not going to keep permanently. We're not going to keep that piece of RNA forever. It's just going to be there to kind of be as a starting point for the build of my new daughter strand. That RNA primer gets laid down by an enzyme known as primase. So that's the circular icon you see there. That's the enzyme primase that's laying down that RNA primer. At that point, the leading strand, or the strand, will build in a 5 to 3 direction. So in this case, the strand starts getting built. Now this is DNA now, not RNA. There's only that little RNA primer at the start. That strand is going to get built with an enzyme called DNA polymerase 3. So DNA polymerase 3 is essentially going to be the enzyme that is going to build the DNA polymer. He's going to lay down all those different monomer groups in order to make a single piece of DNA, which is a polymer, hence the name DNA polymerase, and it's 3. Now, why is it DNA polymerase 3? There's a whole series of polymerase molecules. We're going to only need to know 2 for this discussion today, but suffice to say, the one that actually lays the DNA or the daughter strand down is called DNA polymerase 3. At that point, if it's the leading strand, we're pretty much done. This is going to basically follow all the way until the zipper unzips all the way at the bottom. We're going to have one long continuous piece of DNA of daughter strand being laid down if it's a leading strand. At the end, we have to do one little thing. What's going to happen at the end is this. We are going to have to add one more enzyme called DNA polymerase 1. And we'll have another enzyme that comes along. So I'm going to do this in real time. So what's going to happen is the DNA polymerase 1 is going to remove the RNA primer. So the DNA polymerase 1 lays down a new section of DNA, and the DNA polymerase 1 is my little triangle, my blue triangle. So I'm going to indicate that with my blue triangle. And then that blue triangle is then going to follow down the daughter strand that was just built, and it's going to proofread. It's going to proofread the DNA to make sure that there were no mistakes. Were there any base pairs that were laid down that should have been laid down? Maybe we put a G beside an A, or we put an A instead of a T. That enzyme, this DNA polymerase 1, is going to remove the RNA primer, replace it with DNA, and then proofread the daughter strand that's just been built. There is now only one issue. Where I laid the RNA primer down, and after it was replaced with DNA by the DNA polymerase 1 enzyme, there's a little gap. There's a gap there. There's a little space that we have to fill. So that space we're going to fill in with the last of our enzymes. And that space is going to get filled in by an enzyme known as DNA ligase. So DNA ligase is going to basically seal up any of the gaps that exist between any original locations of RNA primers that were put on that daughter strand to start the process, and where the DNA strand was kind of attached to it. They all have to sort of be sealed up. Now, in the case of the leading strand, there's really only one spot. There was one RNA primer that got laid down at the top of the zipper, and then the rest of that DNA strand just sort of followed down the zipper line. Very easy on the leading strand. Hopefully now you can understand why the dirtbag strand, the scumbag strand, the lagging strand is more problematic, because we're building this sucker in stages. There's all these RNA primers that have to be laid down. So all these RNA primers are eventually going to have to be removed using my DNA polymerase 1. Then I'm going to have to seal up all those gaps using DNA ligase. So there's a lot more work from the enzymes when we get to this lagging strand. Now, there is some terminology that you should be familiar with the lagging strand. There are little sections of DNA that initially get built. As you build it, there's like small sections of DNA that are being built in the opposite direction of the replication fork and the way it's opening. Those little fragments have a special name, and they are called Okazaki fragments. And I'm going to just make sure I spell that correctly. Okazaki fragments, yes, Okazaki fragments, named after the husband and wife team in Japan that discovered them. So these small segments of DNA that are being built, and these fragments only exist on the lagging strand. You will not find them on the leading strand, because again, the leading strand is completely one long piece of DNA, no issues. But here we've got all these, like here's another lagging strand, or here's another Okazaki fragment right there. All these little red sections here, small sections of DNA that are separated by these RNA primers, they are referred to as Okazaki fragments. And each of these fragments are going to have to be sealed up using DNA ligase at the end of the process. So I'll show you what happens on one of these RNA primers, but it's going to happen, there's 3 RNA primers on this side here on the diagram. Again, what's going to happen? Well, the RNA primer is going to get removed using DNA polymerase 1. So we're going to lay down a new piece of DNA. That's there. That's replacing the RNA primer that was there. And that piece of DNA was laid down with my DNA polymerase 1 molecule. That DNA polymerase 1 molecule, as I mentioned, is going to then go and proofread the Okazaki fragment that's beside it, right? Proofreads DNA. And then we've got all these little gaps. We've got gaps here, here. Imagine these are being removed already as well. There's going to be a gap here. There's going to be a gap there. There's going to be a gap there. There would eventually be a gap over here. So all those little black dots that I've got on there, Those are gaps. And each one of those gaps has to be removed and sealed up. Not removed, they have to be sealed using the enzyme DNA ligase. So that's what DNA ligase does. It seals up all these gaps after the RNA primers are basically replaced by DNA using DNA polymerase 1. Okay? So you can see why the lagging strand is much more intensive for the DNA than the leading strand. The leading strand is very easy. The lagging strand, there's also a lot more work involved. And again, this slide is discussing a little bit more about what I discussed about DNA polymerase 1 and ligase. That's on there as well. The final stage, which is termination, doesn't really require any enzymes at all. It's basically now the DNA is sort of opened up and it's got two strands of DNA being built. So here's my DNA, my parent and my daughter strand, my parent and my daughter strand, semi-conservative replication. Now that DNA molecule is going to wrap itself back. They're going to both wrap themselves back into pieces of double helix DNA. Now, at this stage, what we have are two pieces of chromatin that have been duplicated. When they condense at the beginning of M phase, either in prophase or prophase 1, those two pieces of genetic material are going to become the sister chromatids. Those are now the sister chromatids that are attached with that centromere that will then be either split apart during anaphase if it's metaphase 1, or sorry, if it's mitosis, pardon me. They'll be split apart at anaphase in mitosis, or they'll be split apart during anaphase 2 if it's meiosis, right? So this is basically where the chromatids are coming from that we talked about in the cell division unit. Here's that diagram that I just drew in the notes. If you hadn't found it already, great diagram in the notes. There's examples in the textbook as well. There's all sorts of diagrams related to this, and all sorts of sources, but this is a really good one, very similar to what I drew on the board if you weren't able to see all the detail that I was drawing there. Let's do a quick recap of the enzymes that are involved. During initiation, I've only got one group of enzymes at play. Those are my DNA helicases. They're going to unzip the DNA in order to get my replication fork zipping down my zipper, as it were. Elongation obviously has the most enzymes at play. We've got primase, which is going to basically lay down RNA primers, which are starting points for the daughter strands to be built. And remember, all these daughter strands are being built in a 5-3 direction, which is anti-parallel from the parent strand that's beside it. So remember, if you know the configuration of the parent strand, don't get confused because the daughter strand will be the opposite configuration. So if I've got a 3' end of a parent strand here, don't think that the DNA is being built beside it in that direction because that's the parent strand. The daughter strand will be beside it in a 5-3 direction. So the daughter strand is going to get built in the opposite direction. And that's an easy mistake to make. I made that mistake quite a few times early in teaching this course. It's not a mistake that I want my students to make if you can possibly avoid it. But anyways, getting back to the enzymes involved in the elongation, primase again lays down your RNA primers, which are the starting point for your daughter strands. DNA polymerase is going to actually build the daughter strands off the RNA primers. They're basically making the DNA polymer. Once all the DNA has been built, whether it's the leading or the lagging strands, anytime that there is a RNA primer present, there's only going to be one RNA primer present on the leading strand, multiple RNA primers on the lagging strand. We have to remove those using DNA polymerase 1. It will replace the RNA with DNA. Reason for that is, remember, RNA has uracil. DNA doesn't. We have to replace all the uracils with thymines. Otherwise, there's going to be real issues for the DNA and coding off the DNA. So the DNA polymerase will remove those RNA primers, replace them with DNA, and then we'll proofread what DNA polymerase 3 laid down, the DNA that it laid down on the daughter strands. And then finally, any place where you've got an RNA primer and where it was sort of, where these RNA primers were replaced with these new sections of DNA by DNA polymerase 1, we have to seal up the gaps using an enzyme called ligase and DNA ligase. And ligase we'll discuss in great detail in our last conversation in this unit, which will be about genetic engineering. Ligase is used a lot in creating genetically modified organisms. This is how we can take two pieces of DNA, put them together, and then seal them up. It's almost like the glue that holds DNA fragments together. If you want to take two pieces of DNA and glue them together, use that enzyme DNA ligase. That's what it's designed to do. Termination, the recoiling of the DNA. No enzymes really present during that process at all. Hank is going to take his turn here with DNA replication. Watch him. He's going to walk you through the whole process from start to finish. It's about a 10-minute video. Definitely worth your while just to make sure you're comfortable with all the stages that I've referred to today. Your homework for this topic, pretty simple. Grab the question booklet that's in the comments below. There'll be a link to that question booklet. And the question booklet we're going to be dealing with is the molecular genetics DNA replication question booklet, and that'll be due for next day. That's it, folks. We'll be back next time discussing the fascinating process of protein synthesis. Until then, have a good one.